So now that we understand why cells need to divide, why they need to stay small, and that we've learned a little bit about what's inside of them, the chromosomes, and how they are going to be moving around when a cell divides, now we're going to actually learn the how. How does that actually happen? How does a cell split and divide and go from one cell to two cells? And so the essential question we want you to think about today when you take notes on how the cell cycle happens is, can you describe the process? This picture shows a lot of different things that are happening inside of the cell, and hopefully at the end of the video you can come back to this picture and it'll make some sense to you. So in your note guide on pages 9 and 10, we're going to ask you to fill that out, and at times we're going to ask you to draw pictures in that note guide, so you might have to hit pause so that you can get the information down. So the first thing we need to understand is that cellular division occurs in essentially 3 big stages. The first one is known as interphase, and that stage itself has 3 stages to it. This picture here on the right-hand side shows the cell cycle, and this blue part of the arrow shows the first stage known as interphase. You can see that it's pretty big, and a cell spends the majority of its life there. The first part of interphase is known as the G1 stage, and that would be this little piece of the pie right here. This is where a cell grows and gets ready to divide, and because of that it spends most of its life there. The second part of interphase is known as the S phase, which is right here, kind of in the middle, and the S stands for synthesizing. Synthesis means to make something, so in this stage we're going to be making something, and we'll tell you what that is in a little bit. The next stage is the G2 stage right here. Again, the cell is going to grow and do any last-minute changes it needs to before it actually starts to divide. And that division process is known as mitosis. This is probably what you learned in middle school. It has 4 phases to it, and we're going to review those today. And the final one, which is just this tiny little sliver right in here, is known as cytokinesis. If we look at this word backwards, cis is one of our roots, which means the action or process of. Kine means to move, and cyto refers to cell or cytoplasm. So cytokinesis is the action or process of moving the cytoplasm. Essentially what we're saying is we're going to take that cell and divide it into 2 new cells. And so the first part is called interphase, known as the G1 phase, which is the first part of interphase. And as I stated before, this is where a cell does most of its growing. Also, some new proteins and organelles are made. And so it's an important process to get the cell physically ready to start this division process. The next stage is known as the S phase. And as I stated before, the S stands for synthesis. Well, if we're going to synthesize something, we're going to make something. What are we making? That's DNA. And so in this stage, those chromosomes that we talked about yesterday are replicating. That chromosome, which looks like half of an X, is going to replicate. And so it's going to make that characteristic X shape. That's when that happens right here in the S phase. Here's another picture of what I'm talking about. This could be considered a chromosome, and when the material doubles, it makes an exact copy of itself to give us this picture over here on the right. Some geneticists and people would say, well, this is a chromatid, and then the chromatid multiplies. You could say that as well, and as a result, these 2 pieces are sister chromatids. Scientists use the terms interchangeably, but technically we would say that this is a chromosome, even though it's half of one. The final part of interphase is G2. This is where organelles for cell division are produced. Again, the G stands for growth, and specifically the G2 stage is right before mitosis, and so we're going to replicate or grow more mitochondria. If you think back to our cell unit, we said that the mitochondria is what gives a cell energy. The cell is going to need energy in order to carry out cellular division, and so it's going to multiply and grow as many mitochondria as it can to get ready for the next stage. Here's my attempt at being funny today. What did the biologist say when his sister dropped a beaker on his foot? Ouch! My toe, sis. Get it? The second stage of cellular division is called mitosis. This is a process where the nucleus, the control center of our cells, divides that nuclei into two, and you get the same number of chromosomes in each new cell. That new cell has the same number of chromosomes, so it's not going to be any different than the original cell. It's going to have the same kind of chromosomes. So essentially what we're making are identical cells to repair or replace ones that have gotten damaged. And there's 4 different phases to mitosis. I tell my students to try to remember the acronym PMAT. The P stands for the first phase, prophase, M for metaphase, A for anaphase, and T for telophase. So here's where you're probably going to want to hit pause and try to get these pictures or diagrams in your notes. There should be circles on your note guides. That's going to represent the cell or the cell membrane, and you need to include different things in your picture. For prophase, some things that happen within the cell to get it ready to split and divide is that the nuclear envelope, which is another way to say the nuclear membrane that surrounds the nucleus, starts to break down. So we see these little break points here. That's the nucleus starting to break down, or nuclear membrane. We also have these spindle fibers that are starting to form. These spindle fibers are what we're going to use to pull chromosomes apart. And so they have a really important job because they have to make sure they pull our chromosomes apart correctly. In this phase, under the microscope, you will be able to see the chromosomes. There are these dark purple spots right here, and you can see them because the chromosomes have already doubled. So in your notes, we want you to draw the following organelles in here: the nuclear envelope or membrane, the spindle fibers that I mentioned before, the duplicated DNA, or another way to say that would be the chromosomes. Label the cytoplasm. Remember, that's the fluid-filled part within the cell. And then these yellow structures that look like 2 cylinders, those are called centrioles. And those centrioles are proteins that actually make these spindle fibers so that they can pull the chromosomes apart. The second stage is known as metaphase. In metaphase, simply what happens, meta is one of your roots for this unit, which means middle. So they're going to line up in the middle of the cell so that one sister chromatid is on one side, one sister chromatid is on the other. The spindle fibers are going to eventually attach to those chromosomes, and those spindle fibers attach at the centromere. Remember, that's the center part of the chromosome. And so here's a picture of what it looks like under the microscope. You can see the spindle fibers are these darker pink lines, and the chromosomes in the middle are the darker purple ones. Take a minute and in your notes hit pause and then draw in the spindle fibers, the duplicated chromosomes, and the centromere in the middle. It looks here like in this picture they've drawn 2 centromeres. It really truly should just be 1. In anaphase, what happens here is that the cell is getting ready to pull the material apart. And by material, I mean the genetic material, the DNA. The chromosomes get split into chromatids, and one chromatid is getting sucked back to the centrioles on one side of the cell, and the sister chromatid is getting pulled back, almost like a lasso, getting pulled back here to this centriole. So this is where the spindle fibers have a very important job to make sure an entire chromatid goes to each side of the cell. If unfortunately the spindle fiber pulls the entire chromosome, both these chromatids to one side, that's when we could potentially have issues within the cell. So again, here's a picture over here on the left of what it would look like under the microscope. You can see these chromosomes are getting dark, and this is one chunk of all the chromatids on one side, and here's the other chunk over here. So again, hit pause and label the spindle fibers, draw in your chromosomes, and label the centromeres. The last phase of mitosis is telophase. Telo or telo refers to the end of something. In this stage, we're starting to end the division process, and we're going to eventually get 2 cells out of this. And so the cytoplasm starts to divide and pinch in, And you can see that here on both sides of the cell. The nuclear envelope or membrane starts to reappear. That's what these white structures are. And then the chromosomes start to uncoil. They're no longer condensed. Under the microscope, you can actually see this indent. And in animal cells, we call that a cleavage furrow. I know it's got a goofy name, but that's what we call it. So again, stop the video and label the different parts within this phase on your note guide. The last part of cellular division is known as cytokinesis. This is where we actually end up completely dividing the cytoplasm right here to form two new cells. And so the chromosomes have decondensed. And so cytokinesis is really important because it's here where the cell literally goes from one cell to two cells. And so in your notes, you can simply just draw two cells that have started to form. We want you to label the cytoplasm. That should be easy for you at this point. The decondensed chromosomes, these dark purple structures, and that you see over here as well. Here's the nuclear envelope that's reappeared, and then you should be set. So with mitosis and cytokinesis, these two stages are what produce new identical cells that allow an organism to simply grow. That's how we grow. We don't just get bigger and bigger in terms of our cells getting bigger. They're splitting and dividing, and this allows our bodies to replace or repair anything that has gotten damaged, whether you get injured in a match or a game, fall off your bike or skateboarding. And this process is what also allows some organisms to reproduce asexually. Remember earlier in the week we were talking about different ways in which organisms can reproduce. A cell can actually do this on its own. And so there's a few small differences we want to talk about between plants and animals. In an animal cell, cytokinesis happens when the cell goes from 1 to 2, which is the same for plant cells, but in animal cells where the cytoplasm literally starts to break off from each other, we call that a cleavage furrow. And so it's different in plants. When plant cells start to divide, they get this line right down the middle, and we call it a cell plate. And you can see that under the microscope as well, which is what we're going to do tomorrow, and you will see these differences occurring. Here is a picture of a plant cell with all the chromosomes lined up in the middle. That's metaphase. We've got other pictures over here to the side where you can see the nucleus. That would be interphase. Here's another cell where you can start to see the condensed information. That would be prophase. And so we're going to give you some practice identifying some of these different cells in different stages. And so here's another picture from your textbook that shows a cell going through all of the different phases. And there is a video clip here for you to watch if you have any questions. Tomorrow we're going to review and go over these steps and stages, and you will be able to ask any questions you have of me about this. Otherwise, you can look in your textbook too, in chapter 10, that goes over and reviews this information. All right. Thanks and have a good day, guys.